Hello everyone and welcome to our video demonstration of Experiment 3, Protein Extraction and Experiment 4, Uv-Vis Spectrophotometry for BIO552, Analytical Biochemistry. Now, let's move to our team member introduction.
Protein extraction is the process of isolating and purifying proteins from biological samples such as cells, tissues, or microorganisms. It is an essential first step in many biochemical and molecular biology experiments because proteins must be extracted before they can be identified, quantified, or analyzed. The choice of extraction method depends on factors such as the types of sample, the target protein, and the attended downstream applications. In these experiments, proteins were extracted from Pseudomonas aeruginosa cells using a radioimmunoprecipitation assay, lysis buffer, followed by centrifugation to separate soluble protein from cell debris. The extracted protein were then stored for subsequent protein quantifications using Uv-Vis Spectrophotometry through the Bradford assay.
Objective for Lab 3 protein extraction are to extract protein from Pseudomonas aeruginosa cell using RIPA lysis buffer. Second, to obtain a protein extract suitable for downstream protein analysis. And third, to prepare the extracted protein sample for protein concentration determination using the Bradford assay in the subsequent experiment.
The materials include Pseudomonas aeruginosa culture, RIPA buffer, centrifugation, microcentrifuge tube, micropipette, ice and icebox.
Now let's continue with procedures. Microcentrifuge tube were labeled. 1.5 ml of bacterial culture Pseudomonas aeruginosa was loaded into a microcentrifuge tube. Next, the culture was centrifuged at 10,000 rpm for 5 minutes and the supernatant was discarded. Then, 0.5 ml of RIPA lysis buffer was loaded into microcentrifuge tube. The pellet was resuspended and the microcentrifuge tube was immediately incubated in an ice box for 20 minutes. The mixture was centrifuged at 10,000 rpm for 5 minutes and the supernatant was transferred to another microcentrifuge tube. Lastly, the microcentrifuge tube containing the supernatant was stored at -20°C until further use on Experiment 4 Uv-Vis Spectrophotometry.
degradation was used to separate cell debris from soluble proteins based on differences in density, resulting in a pellet at the bottom of the tube and protein containing supernatant above. During cell lysis, the protein sample was kept on ice to reduce protease activity and prevent protein degradation to maintain protein stability. If the protein sample is too diluted, it can be concentrated using protein precipitation or by reducing the extraction volume before analysis. The repalysis buffer consists of 3 HCl to maintain pH and HCl to maintain ionic strength, and P40 or triton X100 and sodium deoxycholate to disrupt cell membranes, STS to solubilize proteins, ADTA to inhibit metal-dependent enzymes, and protease inhibitors to protect proteins from enzymatic degradation.
Protein extraction successfully isolated soluble proteins from Pseudomonas aeruginosa using RIPA buffer. This extract protein was stored for subsequent experiments using the Bradford assay.
Uv-Vis Spectrophotometry is an analytical technique used to measure how much ultraviolet or visible light is observed by a sample. It is widely used to determine the concentration of substances in a solution. In these experiments, the Bradford assay was used to measure protein concentration. The assay uses cool massive brilliant blue dye which binds to proteins and changes from brown to blue. The intensity of blue color is measured at 595 nanometer, and the absorbance is used to determine the protein concentration by comparing it with a standard curve prepared using bovine serum albumin (BSA).
Objective for Lab 4 Uv-Vis Spectrophotometry are to prepare a BSA standard curve using the Bradford assay. Next, to measure the absorbance of protein samples using a Uv-Vis spectrophotometer at 595 nanometer. And lastly, to determine the concentration of unknown proteins samples using the standard curve. The materials include spectrophotometer, micropipettes and tips, microcentrifuge tube, cuvette, 1 mg per milliliter solution of bovine serum albumin, Bradford region, protein extract from lab 3, and 2 unknown concentration. And lastly, acetone.
Step 1. 5 1.5 milliliter tubes were labeled with number 1 to 5 respectively. Step 2. 0.8, 0.6, 0.4, and 0.2 microliter of distilled water were added in tube 1, 2, 3, and 4 respectively. Step 3. A range of standards containing 0.2, 0.4, 0.6, 0.8, and 1 mg per milliliter of bovine serum albumin were prepared as stated in the table. Step 4. Another 10 microcentrifuge tubes were obtained, labeled, and were added with 990 microliter of Bradford reagent to each of these tubes. Step 5. One of the tubes was added with 10 microliter of distilled water as a blank. Step 6. 5 tubes containing the Bradford reagent were added with 10 microliter of each of the BSA solution prepared in Step 2. Step 7. 2 tubes containing the Bradford reagent were added with 10 microliter of each of the given unknown concentration, which is unknown A and B, and lastly, the 2 tubes containing Bradford reagent were added with 10 microliter of each of the protein sample prepared from Lab 3. Step 8. Each tube was capped and inverted 5 times to mix, and the sample were allowed to stand for 5 minutes. Step 9. The blank end samples were poured into a cuvette after 5 minutes. Then, the cuvette contained blank was put in the spectrophotometer and 0 at 595 nanometer. Once the absorbance reading is 0, the cuvette was removed, and then the next sample with the lowest concentration was put in the spectrophotometer, and the absorbance was read at 595 nanometer. Step 10. The absorbance was recorded in a table. Then the steps were repeated until the highest concentration sample was read. Step 11. The blank and other sample solution were disposed in a waste container after completed. The cuvettes were cleaned properly and the spectrophotometer was switched off. Step 12. A standard curve was plotted using an Excel using the data recorded.
So this is our results. BSA standard curve was prepared by measuring the absorbance of protein standard at 595 nanometer, producing a linear curve with an equations. The concentrations of unknown A and unknown B were that they obtained from the spectrophotometer For sample 1 and sample 2, only absorbance value were measured and their protein concentrations were calculated using the equations of the standard curve.
The Bradford assay successfully determined the protein concentration because the cumulative brilliant blue dye binds to proteins, producing a blue color whose intensity increases with protein concentration. The Bradford assay was used to determine the protein concentration of the unknown samples by measuring their absorbance using a Uv-Vis spectrophotometer. The protein concentration were calculated by substituting the absorbance value into the linear equation obtained from the BSA standard curve. Based on the standard curve equation, the protein concentration of sample 1 is 0.33 mg per millimeter, while sample 2 has a protein concentration of 0.28 mg per millimeter. Although sample 2 produced a slightly negative absorbent value, the calculated concentration remained positive because the equation had a negative y-intercept. The standard curve generally showed that the absorbance increased with increasing BSA concentration, demonstrating the positive relationship between protein concentration and color intensity of the Bradford assay. However, the graph was not perfectly linear because the absorbance value for the standard 4 was lower than standard 3. This inconsistency suggests that experimental error occurred during sample preparation or absorbance measurement, which reduced the accuracy of calibration curve and affect the calculated concentration of sample 1 and 2. Several factors may have contributed to the errors observed in this experiment. The slightly negative absorbance value obtained for standard 1 and sample 2 most likely resulted from minor instrumental noise, incorrect blank calibration, or baseline drift in the Uv-Vis spectrophotometer, rather than indicating the absence of protein. In addition, pipetting inaccuracy, dirty or scratched cuvettes, fingerprints on the cuvette surface, air bubbles, incomplete mixing of the Bradford reagent with the samples, or improper cuvette positioning may have affected the absorbance readings. These errors may explain the irregularity observed in the standard curve and the slight deviation in the calculated protein concentration of sample 2.
So in conclusion, the Bradford assay and Uv-Vis spectrophotometry successfully measured the concentration of the extracted protein sample by using its absorbance at 595 nanometer with a BSA standard curve obtained from the graph.